Brilliant. Thank you very much, Nikki. Well, delighted to now introduce Dr. Monica Gamez from Bristol. Monica did her PhD, actually finished it quite recently, so congratulations, Monica, with Dr. Becky Foster in Bristol, and now she's secured a postdoctoral position with the same group, and she's going to talk to us about the endothelial glycocalyx. So over to you, Monica.
Thank you. Can everyone see this all right?
Yeah, we're great.
Okay. So yeah, thank you, Nina. Like she said, my name is Monica Gamez. I'm a postdoc at the University of Bristol. I actually haven't done my Viva yet, so I'm not officially a doctor.
Apologies.
But it's coming up. It's about 10 days from now, so hopefully I'll be doctor soon. So yeah, this is some of the work that I did during my PhD, which is work focused on looking at heparan sulfate in its role in the endothelial glycocalyx and how we can therapeutically target it during diabetes. Okay, so first I'll give some background on the endothelial glycocalyx, or E-glycocalyx. This is a schematic of the glomerular filtration barrier, and the E-glycocalyx is a carbohydrate-rich layer which lines the luminal side of endothelial cells found throughout our vasculature system. It's primarily composed of proteoglycans and glycosaminoglycans, or GAGs, the main ones being chondroitin sulfate, hyaluronic acid, and heparan sulfate. And of these, heparan sulfate has been shown to be the most abundant sulfated GAG found in the glycocalyx. So the glycocalyx serves as a selective barrier based on both size and charge. And we've shown, as well as others, that the glycocalyx is damaged during diabetes. So we've shown that the kidney E-glycocalyx volume is reduced in diabetic rodent models. And we've also shown that glomeruli from diabetic rodents have a significant increase in glomerular albumin permeability. So the glycocalyx basically serves as an important barrier in maintaining vascular permeability. Oh, that's not one. Okay. So as I mentioned, heparan sulfate is the most abundant GAG, and heparinase, which is an enzyme that degrades heparan sulfate, has also been shown to be upregulated during diabetes. So in type 2 diabetic patients, it's been shown to be systemically upregulated in the plasma and urine, which is shown in these graphs. It's also been shown in diabetic nephropathy tissue that heparinase levels are elevated compared to normal tissue. And there's also a diabetic model which shows that heparinase knockout mice fail to develop diabetic nephropathy compared to the wild type control. So this is showing that heparinase plays an important role in the development of diabetic nephropathy. So this leads me to the hypothesis that preventing heparan sulfate degradation by inhibiting heparinase will help to reduce the loss of E-glycocalyx and the development of the associated microvascular complications during diabetes. So to test this hypothesis, I took two approaches. The first was by showing that HS is present in the glycocalyx and that it contributes to glomerular E-glycocalyx structure and function in healthy mice. And the second was to demonstrate that treatment of diabetic mice with a novel heparinase inhibitor will help to prevent E-glycocalyx damage. So this first approach has kind of two further segments. So I used enzymatic removal of heparan sulfate to test the importance of heparan sulfate. And then I also used a transgenic mouse model which had reduced endothelial HS biosynthesis. Okay, so first I'm just showing that heparan sulfate is present in the glycocalyx. So this is a mouse glomerulus here. Green staining is heparan sulfate. You can see here the magnified image of a single capillary. You can see heparan sulfate staining here on the lumen side of the capillary. So that's suggesting that heparan sulfate is present in the E-glycocalyx. And then moving on to the enzyme studies. For this, I used FBB mice, and they were treated with either heparinase, inactive heparinase or active heparinase, which degrades heparan sulfate. And then 30 minutes later, I followed that with perfusions with Ringer or Alcium blue for electron microscopy studies and permeability studies. I'll quickly run you through the permeability assay that we use since I use it throughout the talk. This assay was developed in our group, and the way that it's done is we isolate individual glomeruli from mice which have been perfused with fluorescently labeled albumin. And then we measure the change in fluorescent intensity in individual capillaries. And it's this change in fluorescent intensity which allows us to calculate apparent glomerular albumin permeability. Okay, so now looking at the E-glycocalyx in these studies, these are representative images of the glomerular filtration barrier. So this dark staining here found on the endothelial cell is the glycocalyx. And when I quantified these images, I found that there's a significant reduction in E-glycocalyx depth and coverage in active treated mice, and podocyte glycocalyx was not affected. And then I also looked at permeability in these mice and found that they had a significant increase in glomerular albumin permeability when treated with active enzyme, showing that heparan sulfate plays an important role structurally and functionally in the glycocalyx. Okay, so now we move on to the transgenic mouse model that I mentioned. This is a schematic of HS biosynthesis, and you can see one of the first things that occurs is HS chain formation. And this is accomplished by HS polymerase, which is made up of two separate enzymes, EXT1 and EXT2, and both of them are required for HS chain formation. So we have a mouse model which allows us to specifically knock out endothelial EXT1 using the LOX-P or Cre-LOX-P system by treating the mice with doxycycline. So for these studies, to ensure that the model was working, we treated mice with doxycycline for 3 weeks to induce the knockout. And then we isolated glomerular endothelial cells by FACS and then looked at EXT1 mRNA. So you can see here, there is a significant reduction in EXT1 mRNA in the EXT1 knockout mice compared to littermate controls. So we know that the model's working. Okay, so now looking at the glycocalyx in these animals, again, we have glomerular filtration barrier images. You can see the glycocalyx here nicely on the littermate control, and then EXT1. Quantifying these images, I found again that there was a significant reduction in E-glycocalyx depth and coverage in the EXT1 knockout mice. And again, podocyte glycocalyx was not affected. And then looking at the permeability, I found again that there was a significant increase in glomerular albumin permeability in the EXT1 knockout mice. So this is just another way of showing that heparan sulfate plays an important role in the glycocalyx. I also looked at albuminuria in these mice, which is another readout of glomerular function. And surprisingly, we did not see a significant increase in the EXT1 knockout mice. So we thought about what might be going on here, and one of the things that regularly occurs is tubular reabsorption, and it's only when the system's overwhelmed by albumin that you start to see those changes in albuminuria, such as in diabetic nephropathy. So the idea behind this was that tubular reabsorption was masking small changes that occur in the EXT1 knockout mice. So to test this, again, we used littermate control and EXT1 knockout mice, treated with doxycycline to induce the knockout. And then I collected a pre-treatment urine, and I immediately treated with lysine, which is known to be a potent tubular reabsorption inhibitor. And then I collected a post-treatment urine. And then I calculated the fold change between pre-treatment and post-treatment and compared the two for both mouse groups. So you can see here, there's a significant increase in fold change in UACR in the EXT1 knockout mice compared to littermate controls. So this is suggesting that there are changes in albuminuria in the EXT1 knockout mice, but they're small, so they're being masked by tubular reabsorption. Okay, so now that I've shown you that heparan sulfate is important in the glycocalyx, we then wanted to test whether or not we could therapeutically target it during diabetes. So for this, I used a type 2 diabetic mouse model. So I used DB mice. And I had 3 groups, leans treated with vehicle, diabetics treated with vehicle, and diabetics treated with the novel heparanase inhibitor. And the treatments began at 9 weeks of age. They were given daily injections, and then that was for 2 weeks, and at the end collected urine and did the perfusion and tissue collection for analysis. As I mentioned, this is using a novel heparanase inhibitor. So the currently available inhibitors are all linear polymers, and this inhibitor is a dendrimer. So they have the added benefit that they're inexpensive to produce. They're easier to have a quality control due to the synthesis method. And then they also, this inhibitor specifically has been shown to have nanomolar potency and no significant anticoagulant activity. Okay, so looking at the glycocalyx in these studies, again, you can see the glycocalyx here on the endothelial cells. And quantifying these images, I found that there was a significant reduction in E-glycocalyx depth and coverage, which we've previously shown. But we also saw that mice treated with the novel inhibitor had a significant increase in E-glycocalyx depth and coverage. So that's suggesting that heparinase inhibitor is having a restorative effects on heparan sulfate in the glycocalyx. And again, podocyte glycocalyx was not significantly impacted. And then looking at glomerular function in these studies, we found that albuminuria was significantly increased in the diabetics, which we would expect. However, it was not significantly increased when the inhibitor was given. And then using the permeability assay, which is a more sensitive and direct measurement of glomerular albumin permeability, we found that there was a significant increase in the diabetic mice, and that was significantly reduced when the inhibitor is given. So it's suggesting that the heparinase inhibitor is not only having restorative effects on glycocalyx structure, but also function. Okay, so just to summarize what I've talked through, hopefully I've convinced you that heparan sulfate plays an important role in the glycocalyx structure and function, and I've shown that in two different ways. The first was using enzymatic removal of heparan sulfate, and that showed that there was E-glycocalyx damage and increased glomerular permeability. And the second, using the transgenic mouse model that had reduced HS biosynthesis, and that resulted in glycocalyx damage as well. And then I showed that heparan sulfate can be therapeutically targeted by inhibiting heparinase in a diabetic model, and that resulted in restorative effects on E-glycocalyx structure and function. So I'd just like to thank everybody that was involved in this work, my funding, and thank you to Nina and Dean for giving me the opportunity to speak at this conference. I mean, I'll take any questions. Thank you.
Thank you, Monica. That was a really clear and lovely study with some nice translational implications as well. Are there any questions? Oh, there are questions rushing in. We've got a question from Casper, and he says, do you see a correlation between albuminuria and the level of glycocalyx loss?
So I've not looked at albuminuria and E-glycocalyx loss. I have looked at glomerular albumin permeability and E-glycocalyx depth and coverage, and there is a significant correlation with those. So yeah, this doesn't prove that E-glycocalyx is responsible, but it's certainly suggestive that it is.
Yeah. There's a follow-up question from that. How much loss is needed? Is height or coverage percentage the most important parameter?
Yeah, that's a good question. I don't know which one. I don't think anyone knows whether or not it's height or coverage. When you think about it, you might think coverage is more important because you're going to have these gaps where stuff can kind of get through. But I don't know in terms of an actual value. I don't know. This is using electron microscopy, which obviously is not going to give you the true thickness of the glycocalyx. Which, yeah, I guess you can also do it using immunofluorescence. You can look at the thickness of glycocalyx that way. But I don't know if the actual values have been calculated in terms of how much is actually required to see changes.
Good. There's lots of questions. I'm going to jump to one at the bottom that's just come in from Giovanni. Perhaps I can encourage people to come and show themselves and ask their questions and not to be shy, because it'll be nice to see so many of our members face to face. But here's a question from Giovanni. Have you assessed permeability of smaller molecular markers, two different markers, for example, to assess changes in pore size of the fenestrated capillaries?
No, we haven't. At least not for my project. That wasn't the focus. I just looked at glycocalyx, but didn't focus on, for instance, like changes in fenestrations, that kind of thing.
I'll take one final question then from Sebastian. Is EXT2 a gene isoform? Would you see bigger effects with a double knockout?
So both of them, there have been studies done that both of them are required. So even though they both have activity, knocking one out is sufficient to get rid of HS chain formation. I don't know actually if you knocked out both. I suppose you would expect that you might get a better knockout. I mean, these are conditional knockouts. So if you can't just knock out, they're embryonic lethal. So it has to be a conditional knockout. But yeah, you might be able to knock out both and get better knockdown. Yeah.
Well, there's a lot more questions for you, Monica, but I'm going to send you over online in the interests of time. But well done once again. That was a great presentation. Thank you. And please, if I can encourage people to, actually, since somebody has raised their hand, Zubair, can we see Zubair? And since he's raised his hand, and give him a chance to ask his question face to face.
Hello.
Can we unmute him? Hello, Zubair.
Hi. Thank you. I just wanted to ask, was there any specific stain used for the glycocalyx, and how you quantified the coverage and depth? And we can find variability of depth and coverage within vessels in the same field of view, for example. Did you consider that?
Yeah, so there's kind of, I guess I have two answers for that. So first, I am planning to do heparan sulfate staining on these tissues so that we can visually see changes in heparan sulfate, since what I showed was all EM. So the stain for EM that I used was Alcium blue. So it's basically just a electron dense, positively charged molecule, which binds to the glycocalyx and allows us to visualize the glycocalyx through EM that way. So it's not a specific heparan sulfate stain, but it does stain just general glycocalyx. What was the second part of the question? Sorry.
Well, perhaps actually the second part I'll let you answer online. I think.
Sure, okay.
But thank you very much, Monica,
Great,
and
thank
thank you
you
for coming online.
Thanks.
Thank you. Okay.